Hello there, it's Luke, and in this tutorial about hemostasis and coagulation, we're going to be focusing on platelets, and we're going to be looking at their role in primary hemostasis, by which we mean the formation of a platelet plug, but also the many other roles that the platelets perform in hemostasis generally, and there are lots of them, and I hope by the end of this tutorial you have a healthy respect for platelets. So what are we talking about? Well, here are some cells from within our blood. We've got a red blood cell here, we've got a white blood cell here, they're both quite large, and then down here we have an activated platelet. And you'll no doubt already be aware that in their inactive form, platelets, well, look like little plates spinning around, these sort of small chunks of blood vessels, and when they become activated, they have all these sort of little projections on them that gives them a great surface area, but also helps them to sort of catch one another and stick together. So the sort of primary role, if you like, of platelets is primary hemostasis, is a formation of a platelet plug. So we've got a damaged blood vessel here, and we would be leaking out blood, but for all these platelets clumped together here in this sort of loose, not particularly strong, but good enough for the moment, platelet plug. And here's just a quick recap of the different parts of our diagrams of platelets, red blood cells, white blood cells, endothelial cells on the innermost layer of the blood vessel, all the sort of tissues that sit behind that are supportive, and then clotting factors. So we have our bleeding vessel here. This is the lumen. This is the area of damage. This is where the blood is coming out, and what we want is this platelet plug. So we'll remove the other blood vessels. We'll get rid of the red cells and the white cells so that we've just got our platelets swishing around, and you can see that they're inactivated. They're in their sort of little plate-shaped version of themselves. And into that, we're going to add all of the clotting factors that have been made by the liver. So they're circulating now in the bloodstream. And they're there all the time. These clotting factors are there all the time. It's just for the sake of simplifying the diagram, they weren't in there to begin with. So we've got clotting factors, we've got platelets that have yet to be activated, and we want a platelet plug. Now you'll recall that from the last tutorial we talked about the tissue factor pathway. So when you've got a damaged blood vessel, it exposes this thing called tissue factor to other clotting factors, mainly factor 7, and this sets off a little cascade here of a couple of clotting factors that gives us thrombin, which is an absolutely crucial clotting factor. And one of the actions of thrombin is to activate platelets. So it takes this inactive platelet and turns it into the active form, this sort of spiky version of a platelet that does loads of stuff. There are other things, other substances that will activate platelets, and there's a few of them here, but thrombin is the main one we're going to discuss in this tutorial, and it's the main one I'd like you to focus on. Now when a platelet is activated, as you can see, it changes shape. You've got more surface area for it to interact with things, but also it's more likely to sort of stick to other platelets in this new sort of spiky shape. One of the other things that activated platelets do is they release chemicals from within the sort of stored granules that they have inside of them, and they release something called adenosine diphosphate and platelet activating factor, which, as you can probably guess from the name, activates other platelets. Both these things will activate other platelets and sort of attract them to the site of injury. A particularly interesting thing that platelets do is from their cell membrane, from the phospholipid cell membrane, they produce this substance called thromboxane A2. Again, this is a arachidonic acid pathway process, which will be important to remember when you're thinking about drugs that affect the clotting cascade. And also thromboxane is released by some other cells as well. And why is that significant? Well, thromboxane is a potent vasoconstrictor. So we talked in a previous tutorial about how the blood vessels constrict, which reduces the flow, and not only hopefully reduces the bleeding from the vessel, but also brings all the clotting factors and platelets and other things, tissue factor for instance, in contact with each other to begin that haemostasis, thromboxane will attract other platelets to the site of an injury and then diffuse through the membranes of those platelets and activate them. And the sort of one of the cool things about thromboxane is it has quite a short half-life, by which you'll mean it stops working and degrades very quickly once it's been released. And that's good, because we don't want clotting to be occurring all around the body in the bloodstream. We just want it happening at the site of injury. So the fact that it sort of gets switched off quite quickly is really good, because it means we're not sort of clotting right, left and centre, or activating platelets right, left and centre rather. Another couple of factors that I'd like to draw your attention to are von Willebrand factor and factor VIII. And these circle around the bloodstream. And here we are. So, and they go around together, factor VIII and von Willebrand factor. So we have our damaged blood vessel here. We've got all the different clotting factors and our sort of non-activated platelets. Now, when our platelets become activated, what they do is they'll sort of break that bond between factor VIII and von Willebrand factor. And factor VIII can then go on and get on with its various becoming activated itself and go on with catalyzing other enzymes. And von Willebrand factor sticks onto the platelet itself. And if we imagine these are little sort of concrete trucks, they use their sort of concrete to sort of glue and adhere the platelets to the site of injury. So that's the action of von Willebrand factor. It sort of sticks the platelets to the site of injury, whether that's the collagen or the other sort of fibres and tissues that have been exposed when this blood vessel has been injured. So very briefly, to sum up where we are so far, the activated platelets will change shape. They'll begin to attract other platelets and begin to aggregate, so stick together, group together with those other platelets, and they will then, that clump of platelets will then stick to the injured blood vessel wall using von Willebrand factor to cement themselves there. Now, this is just the picture summing that all up. So we've got our platelet plug formed, and we refer to this first part of haemostasis as primary haemostasis, the formation of that platelet plug. Now, as I said before, hopefully you'll have a new respect for platelets after this tutorial, but they do lots of other things on top of that primary haemostasis. And we're going to mention a few of those now. So first of all, they contain and release some clotting factors. So not only do they make a platelet plug themselves, but they release some of the factors that go to help then support that plug and turn it into a fully functioning clot. They will express something called glycoprotein 2A3B, which really acts like a glue to stick the platelets to the fibrin that is being formed through coagulation, so that the whole thing sort of sticks together and is a nice strong clot. And they also express phospholipids, which when you combine them with calcium, acts as a site for a lot of those enzyme type reactions to occur that need to happen during the clotting cascade. So for instance, we've got this phospholipid on the surface of an activated platelet here, and that provides a space for factor 5 and factor 10 to work together to convert prothrombin to thrombin. As we've mentioned before, thrombin is a really crucial clotting factor for the whole of haemostasis. So here's a quick list of those functions. So it expresses those phospholipids to help to provide a place for other interactions to occur. It has these glycoproteins that stick to fibrin to make a clot. And another thing that we'll talk about further on in our tutorials is that it's actually got actin and myosin proteins that you recall pull together in muscles that will sort of pull the clot together and do something called clot retraction to make the clot stronger. And they'll release other things, including sort of growth factors. So in summary, we said that platelets are activated principally by thrombin, but other mediators as well. And they have many roles within haemostasis. One is the primary haemostasis, so that formation of a platelet plug. They release thromboxane and clotting factors and other things that will attract platelets to the area of injury. and they provide a space on the membrane for other enzyme reactions to occur, so other parts of the clotting cascade to work. Thank you very, very much for listening. Thank you.